Howdy everyone, my name is Aisha Espinosa, and today we're going to talk about caste-specific morphology in Apis mellifera, or more commonly known as honeybees. So based off the articles and experiments I have gathered, my question is how caste-specific morphology is affected by the levels of dopamine produced by queens or worker Apis mellifera. So, dopamine. A little background about it is its levels effect at the metamorphosis stage. It is a biogenetic amine which functions as a neurotransmitter. The neural aspect stems from the fact that dopamine functions as a neuroactive substance that transmits neural signals. So right here you can see in picture A, C1 and C2, and they're highlighted, so this is the portions of the brain they affect in the honeybees. If you go to the next picture where it says neurotransmitters and it shows the brain, and everything highlighted in green is the nerve cord system, and this is everything it's going to affect and everything it communicates with. It also acts as a neuromodulator and neurohormone. The neurohormone it affects the central and peripheral nervous system, and right here you can see the neurohormone, how right here it is in red, and you can see how it travels all the way to the hind area, and you see how it affects and interacts with everything else. Something really strong to note is that the dopamine interacts and has a strong relationship with the metabolic levels. Something noted is that dopamine is continually associated with caste-specific morphology. Dopamine levels are generated in the brain during the pupil development, and this is done via differential expression of genes encoding enzymes involved in the synthesis. A little bit more about the enzymes of genes that help generate and transport dopamine are AMDOP1, 2, and 3. These act as a receptor. So now that we have a little bit more background information about dopamine, we can start to associate it with caste-specific systems. The honeybee's brain is influential up until emergence. The dopamine generated in the pupil brain are dependent on enzymes associated with the uptake. So we briefly did talk about one of them, which is the AMDOP1, 2, and 3 receptor. With caste-specific systems, each vary in dopamine receptors. This influences how much and who produces more dopamine, based off the specific role each bee plays. When it comes to queen bees, they have a higher expression of gene-encoding enzymes involved in dopamine synthesis, but gene-regulated encoding were down-regulated. So dopamine-related genes and regulators differ on the type of honeybee, such as queen bee or worker, and influence their distribution and how they are activated. So right here in the corner you can see this figure, and it shows the AMTH levels, and these levels along with the AMDDC are the biosynthesis and how each are different within the worker and queen. So the queen is in the red, and we can see that the queen has a higher level of the biosynthesis while the worker has a lower level. And then when it came to the AMNAT, this is the dopamine conversion. So we can see the dopamine conversion is just a tad higher in workers than it is in queens. And then the AMDAT is a dopamine transporter, which at first, as you can see, the queen and the worker start at almost the same level, but as they grow on before inclusion, which is before they come out of their honeycomb cell, the queen spikes up until she's an adult. Now, with background information about dopamine, caste-specific systems, how dopamine fluctuates with the synthesis, signaling, and conversion, we can now associate how dopamine actually affects the Apis mellifera. So something that we can see is when a honeybee has higher levels of dopamine, their mandibular shape and index tend to be more noted. The female with the mandibular notch have higher levels of dopamine. So right here we can see that generally the queen usually has higher level of dopamine, and they have a mandibular notch, which can be seen right here, while the workers is more rounded and contains no notch, while the queen has mandibular has a bigger width than the worker. Something else the dopamine affects is the number of ovules. The reproductive workers tend to have higher level of dopamine, and because they have a higher level of dopamine, this is associated with the egg-laying behaviors. Since dopamine affects the ovarian activity, when a honeybee is under a queenless condition, the ovarian development is affected and dopamine levels do spike, creating a wanting to make a more production of queens to help the hive. So when it comes to virgin queens, higher dopamine levels encourage higher levels of locomotive activity, a higher aggression towards other virgin queens. So, if another virgin queen is put in within the same vicinity of one another, they're going to want to fight to be the only queen left, and the workers tend to take them away before anything gets out of hand or they are left without a queen. However, if it does get out of hand and there is a winner, the winner is always going to be the honeybee or queen at the moment that has a higher dopamine level. This is not something that got mass produced, but it is just a level of dopamine they are born with and just is higher in general. We're going to go into dopamine right before emergence. So right here we can see that we have the larval stage, the pupal stage, and the adult stage, and we can see the honeybee exiting the honeycomb cell. Once they exit their cell, they participate in hive activities. Something to note that the reason dopamine is so important is because they influence caste differentiation 2 to 3 days before inclusion. However, this cannot be identified 3 to 4 or even 5 to 6 days before. The caste are affected by dopamine energetic systems, as each caste system differs in their brain. Dopamine changes with age, at the peak of pupal stage, and with receptor subtypes. With this being said, dopamine levels tend to be fourfold higher in the brain of a queen than a normal worker. So now with all the information about dopamine, caste-specific systems, and how dopamine exactly affects an Apis mellifera, and we're noticing that the dopamine levels are affecting at the pupal level and affect how they produce their areoles, their aggression levels, and some features such as the mandibular notch. We can now see how the levels and how high they are when it comes to a queen and a worker. So below are some charts from 6 to 7 days, 4 to 5, and 2 to 3. The levels differ between workers and queen, as you can see. Queens usually have the highest level of dopamine the day of eclosion at 37.247 p-moles per brain, while workers only had their highest level 2 to 3 days before eclosion at 17.197 p-moles per brain. So right here we can see in the middle how they both start. The queen starts and then she dramatically climbs up until eclosion, where she has her highest level of dopamine. Now when it comes to the worker, they stay at a lower level, have a miniature spike, and then as they are getting to eclosion, they have a dramatic downfall. And this can be shown in both A1 and A2, or if you want to compare them at the same time, A3 figure. So we notice that the levels are at least 10.1 p-moles per brain lower when it comes to workers and queens, and that as they get older, queen bees possess larger amounts of dopamine and metabolites in their brain and hemophilites. So in this situation and study that the article did, the queen had 3.6 folds higher of higher level of dopamine than a normal worker. So, all of the information gathered about dopamine, caste-specific systems, how dopamine affects Apis mellifera, dopamine emergence, and the pupil stage levels of dopamine have been gathered from these works cited below. Their experiments were gathered and information was used, and as I based my question off of, I hope everything was made clear. Thank you guys for watching this video. Have a great day. T's and G's.